Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 156th seminar. I call it a Joint Genome Institute or JGI Day. Life is full of ups and downs, or unexpected good luck or bad luck, or are also relative as well. In San Diego, we had multiple days of heavy rain this week. This is very rare, but also this is also might be welcoming if the amounts are not extreme. But in some cities, such as Seattle, more sunny days during especially the winter would be needed. Additionally, the California areas affected by the fire may experience landslide due to heavy rain, showing all are relative. My 2 days, my two trips were canceled, including this week's one, but I spent more time with my lab members and slept for more hours than 5 hours, which is my typical sleep hours. It would have been nice to chair this seminar in the oceanfront hotel room in Panama City Beach, where I am supposed to stay today, but I am chairing it in the earlier morning time in my tiny apartment unit in San Diego. Despite all the difficulties, challenges, and also unexpected events, research and this webinar series must go on. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Dr. Axel Visel. He is a senior staff scientist at Lawrence Berkeley National Lab and also a deputy of science at the DOE Joint Genome Institute. Recently, I met him during the JGI user meeting last year, where he kindly agreed to give a talk for this seminar series. Given his amazing research activities and also service to the research community, I was so delighted to have him, and here he is right now. Axel, thank you so much for your contributions to the research community, and the virtual podium is all yours now. And thank you again.
Hey, thank you, Tesuk, for inviting me here. Good morning to everybody watching live or watching this later on YouTube. Let me share my screen. So as Tesuk just mentioned, I'm at the Joint Genome Institute, and I'm the Deputy of Science there. And for this seminar, for my brief introduction talk here, I first thought about talking about some of my research, which focuses on plant microbial interactions, but then realizing that the main speaker today, Chris, will talk about this in much more detail and probably will have more interesting data to show than I would have. I will not touch on that, but instead, I want to use this opportunity to talk a little bit about the JGI, which I think may be of interest to this audience. So the Joint Genome Institute is located in Berkeley, California. We are located on the main campus of Lawrence Berkeley National Lab. You see our building here. It's a new building, about 5 years old. And we are a national user facility that is funded out of the Department of Energy. So this is one of the ways in which the Department of Energy supports fundamental research in the United States by operating these user facilities. In the case of the JGI, we are focused on genomics, which means we cover research areas such as environmental genomics, bioenergy genomics, and research needed to enable bioproduct research. Now, being a user facility means that we are not per se a research institute, but we are enabling research in the DOE mission space. This means that we offer a variety of capabilities, such as DNA sequencing, DNA synthesis, and anyone can apply to us. So we don't give out grants, but what we do is we provide access to, and free of charge access to these capabilities to scientists around the globe. Importantly, for this audience, this includes a portfolio of DNA synthesis and genome engineering capabilities. Just to give you a broader sense of the kinds of topics that we're working on. So in terms of the research, we focus on themes such as nutrient cycling and functional diversity. We do a lot of data science, building large data resources, building data tools. We also have emerging research areas such as biomolecular materials, biosurveillance, and biopreparedness. We don't do any biomedical research. Unlike many other genome centers in the US, we exclusively focus on non-biomedical research. This means our organisms that we focus on include plants, fungi, algae, and then a lot of microbes, which we study as isolates or as large metagenomes from various environments. At the bottom, you see the various types of technologies and capabilities that we offer. These include DNA sequencing platforms of various kinds, coupling this sequencing with other technologies that we call advanced genomic technologies, which really help to make sense of genome function. This includes, say, single-cell sequencing of microbes, of plant transcriptomes, stable isotope labeling, and so on. We have various metabolomics tools, and then importantly, we have DNA synthesis tools, and I'll talk about those in some more detail. We have experimental capabilities as well as various tools and support mechanisms to help people with genome engineering. And then we have various secondary metabolites focused tools. Just to give you a sense of the scale of our operation, so in any given year, we typically support more than 2,000 users across the globe. The majority of our users come from the United States, but you don't have to be located in the United States. We have users from all across the globe. Really, the main criterion for being eligible to apply to us is not where you're located, but whether or not your science is relevant to the DOE science mission, which means that it's broadly related to environmental or energy questions. You can also see in the bottom left corner that the majority of our users come from the academic space. This reflects our focus on fundamental research. So the primary goal of most of our users is to produce insights, to publish papers, to generate publicly available data sets. But we do have users from industry, from other national labs, from government institutions, and other types of organizations. With respect to DNA synthesis, there is a range of products that we offer to our users. We do fairly conventional small constructs that are a single or a few small proteins in one construct. Typically, many of our projects are large, so it's not just one construct, but dozens or hundreds of constructs at a time. We do a lot of more complicated, larger constructs, pathways, or multiple operons in the same construct. We also work with users in building combinatorial clone libraries. So this is typically applied for purposes such as building biosynthetic gene clusters, where you take ingredients mined from, say, metagenome data sets from various organisms, and you recombine them in various ways to build these combinatorial libraries, to screen through them, to look for outcomes and measure which of them work. We also have tools for building libraries that have a lot of sequence variation in them. And we offer a bunch of strain engineering tools. For the strain engineering, one particular focus area here is the CRAGE strain engineering platform. This is something that we developed a few years back. If you want to know the details, this was published in 2020 in Nature Microbiology. This is a framework where the idea is to take undomesticated bacteria or other organisms and then put the same kind of landing pad into all of them. So you have a phylogenetically diverse range of hosts that can all be conjugated with the same construct. And what this, of course, enables is that you can move from a conventional approach where you have one organism and engineer it to building your pathway or other larger constructs once with the right kind of landing pad compatibility, and then in parallel get it into a range of hosts that you can then characterize in detail. Again, a typical application for this would be, say, expressing a certain pathway in a heterologous manner where you don't know which of your hosts will have the right kind of cellular background to be an efficient producer of whatever product you're interested in. And we've been building this out for various organisms now. At this point, we have in-house fully developed 39 proc species from across 5 phyla that you see here, 3 different classes: Proteobacteria, Actinos, Firmicutes, Bacteroidetes, Cyanobacteria. We also have 2 yeasts that work fully. We are currently working on additional organisms, including more yeasts, more bacteria, and some archaea. Some of these are still in the process of being made fully available for users. Right now, through our standard products, we have 8 different proteobacteria available. The cyanos and firmicutes should come online very shortly, and then the majority of those 39 that we have in-house should come online over the next year or 2 for use in user projects where we make these organisms with a landing pad available to our users. Now, you might wonder how you can take advantage of this, and the mechanism for doing so is to become a user. The way this works is similar to applying for grants, which means you write a proposal to us. This proposal gets reviewed by a review committee that is composed of external reviewers, and then the highest scoring proposals get supported by the JGI. We have multiple user programs that have deadlines around the year. On this slide, I've listed the 3 that are most relevant to the synthetic biology community. The first one at the top is the so-called Community Science Program Annual Call. These are the largest programs that really provide support for large projects. Often it's not a single investigator, but an entire consortium of investigators that apply for these. It's mostly for genomics capabilities such as sequencing, but it also supports projects that combine sequencing and DNA synthesis. The deadline for this one is coming up in April. For those of you who are early career investigators or who haven't worked with the JGI before, we have a smaller call that is called the New Investigator Call. This has a deadline in the fall, and it again offers access to all the products that we have, including DNA synthesis and all of the sequencing products. This is specifically designed for people who haven't worked with JGI before. You don't have to be early career. The only criterion is that you haven't been a JGI user in the past. And the bar is a little bit lower for these. So oftentimes people use these to generate preliminary data that can then provide the basis for a big annual call proposal. And then finally, purely focused on our functional genomics capabilities, we have the functional genomics call. The deadline for this year has just passed 2 months ago, but we have another coming up in early 2026. This is the call if you have DNA synthesis only or strain engineering only requests. So I encourage you to look into that if you're working in this space. You don't have to wait until the deadline approaches. If you have ideas for potential projects, please get in touch. And the best people to contact are shown here on this slide. Yasu Yoshikuni and Ian Blady. If you have science questions, so for example, if you have an idea for a science project and want to discuss, for example, if it would be well aligned with the DOE and JGI science mission, Yasu is the person to ask. He's the head of our DNA synthesis science program and provides scientific support for all of our user projects. If you have questions about the specific technology that we offer, for example, the feasibility of your project idea, or if you want to get more information about what exact organisms we have available right now or what organisms are expected to become available shortly, then Ian is the person to get in touch with. He's the lead of our DNA synthesis platform, so he oversees the actual production activity. With that, I want to thank all of you for your attention. We're not taking questions right after my talk, but at the very end of the session. Thank you.
Thank you so much. I mean, I also echo, also encourage people, audience to apply. Actually, myself also benefit from JGI, this program. We are actually asking the first sequence to be synthesized, and actually we got up to 500 kilobase of DNA synthesis. That's a lot of DNA. And then we are very excited because we identify some plastic degrading community samples. But the problem is we need to identify the individual sequence. So we are kind of guessing some of that, and then we are asking the JGI to synthesize for us, and then we're gonna test those things. So, this is one example, I can give, but if you have any idea, but you need some sequence, free sequence or free DNA, this is kind of one, avenue you can think about. So thank you so much. This is amazing opportunity, amazing service to the community, and I really appreciate it. Okay. Now the main speaker of today with a longer introduction, Dr. Chris Dundas is an assistant professor at the University of Georgia, where he holds a joint appointment between the Department of Plant Biology and Institute of Bioinformatics. In 2015, he completed his bachelor degree in chemical engineering at the State University of New York, or SUNY Buffalo. As an undergraduate student, he performed protein engineering research with Professor Sheldon Park to tune the structured-biotin interaction for bioseparations and cell biology applications. In 2020, he received his PhD in chemical engineering as well at the University of Texas, Austin, under the guidance of Professor Keith. As a graduate student, he developed synthetic biology tools for electroactive soil bacteria to control redox-driven inorganic and organic material synthesis. From 2021 to 2024, Chris was a Tomcat postdoctoral fellow in sustainable energy at Stanford University in the lab of Professor Jose Diney. There, he created biosensors in root colonizing bacteria and synthetic metabolic circuits in transgenic plants' roots to measure and modulate soil carbon accumulation. In summer of 2024, last year, he started his independent career at the University of Georgia, where his lab focuses on using plants and microbial synthetic biology to engineer plant-microbe environment interactions for agriculture and sustainability technologies. Chris, thank you so much for your time today, and please take it away. All yours now. Thank you.
Thanks so much for the introduction, Tae-Sok, and the opportunity to speak to the folks here in the SynBYSS seminar. I've always really enjoyed watching them over the years now, and it's been just, I think, a great way to kind of connect the synthetic biology community together. And it's also really nice to hear Axel as the person before me. I've been such a fan of the JGI resources, and I guess I first got acquainted with them when I was out at Stanford with my postdoc. Went to the user meeting a few times and was part of one of the CSP calls. So I also echo that it's a fantastic resource to basically just get free DNA sequencing and synthesis. And certainly for young professors and more senior faculty and other scientists, I think it just is a really awesome community that I encourage folks to get involved with. So today I'm going to talk about a fair bit of my work from my postdoc, and it's really kind of a lead-in to some of what my lab is doing. And like Tae-Sok mentioned, my lab and work is focused on how we can build genetic circuit tools in both microbes and the plants that they colonize. And so I'm going to really be looking at how we can use these types of tools to analyze the exchange of carbon, mainly in the direction of coming from the plant host and going to microbes. Okay. So yeah, just to give a bit of a 10,000-foot view overview of my lab, I'm really interested in just how plants are interacting with the extracellular world, both microbes and the other abiotic factors that are out there. And carbon is really one of the main ways that plants are able to talk to the world outside of themselves. So we all know that plants can capture carbon through photosynthesis. That's what fuels the biomass growth demands that plant tissues have. But the cool thing is they actually release a lot of this carbon back into the environment, where it can serve as what drives the formation of soil organic matter, but also it is quite readily received by the microbiota that is colonizing the above ground parts of the plants, we call that the phyllosphere, as well as the below ground parts of the plant, which we term the rhizosphere. And so what's currently not really known at present is what are the types of molecules that are being released from plants into the microbiome? What are the types of molecules that the microbiome is giving back to the plant? And how does this affect the interaction between these two kingdoms of life? And perhaps from a technology standpoint, how might we harness these interactions to make healthier plants or soil systems that are more enriched for the right types of microbes? Can we use this to fortify our agriculture and other types of bioproducts relying on crop systems? And so kind of a big idea of my group is that synthetic biology is really this super enabling tool to dissect and engineer these processes. So the types of questions we ask are, well, what types of metabolites are plants and microbes exchanging back and forth? So it's kind of a fundamental science question. But then the applied question is, well, can we engineer maybe some of these keystone carbon fluxes using circuits in either plants or microbes to make plants that maybe can access nutrients like nitrogen and phosphorus more readily? Can they withstand things like drought stress, which are becoming increasingly prevalent? Can we make plants that secrete more carbon into soil to enrich the soil carbon stocks of a lot of our agricultural lands where we've depleted the soil carbon that's present? And kind of, I think some of the more emerging technologies are, can we use the rewiring of carbon fluxes with synthetic biology to domesticate new microbes and plants that have been previously outside the realm of what we've been able to do genetic circuit design in? So my background is more so in microbial synthetic biology, and like Axel was mentioning, there's now increasing tools to domesticate non-model microbes. I'm going to make the argument that we can also use these types of plant and microbial carbon cycle synthetic biology tools to also domesticate non-model plant species as well. And I think there's a lot of exciting opportunities in plant synthetic biology along those lines. Okay, so this is kind of the big picture goals, but why is it tough to try to engineer plant and microbe carbon cycles? Well, right at the outset, it's really tough to actually measure what plants are secreting out into the environment. So I'd say at present, we really don't have a good spatiotemporal understanding of what types of metabolites are plants secreting, mainly I think of the root systems primarily, and where exactly are they coming from. So if carbon was secreted at the root tip versus the more mature parts of the plant, we really don't have a sense of what are the drivers and compositions of these types of metabolites that plants are secreting, say, into soil systems. In parallel, we don't understand the genetic underpinnings that drive the release of these metabolites. But I'm a synthetic biologist, so we always ask, well, what are the control points? What genes or regulatory systems can we dial to tune these types of compositions or fluxes? We also currently lack that type of understanding. Similarly, we don't understand for the microbiome that is inhabiting these root systems, what are the control points in the microbes? What genes or regulatory systems can we rewire to affect how microbes are using carbon that plants are giving them, and how that could be controlled to regulate the composition of what microbes get to grow, which ones don't, and the activity of these microbes to maybe make them more beneficial for helping plant or soil health. And so a key hypothesis of my group is that we can use this idea of trans-kingdom genetic circuit design to understand and engineer these processes. And so what do I mean by that? Well, what I mean is we can create transgenic plants where we can systematically dial the expression of plant genes that might be involved in releasing carbon to the environment. And then we can use engineered microbes as the super fast microbial readouts to let us know the success or failure of our plant synthetic biology efforts. And so what I mean by that is we can use the last 25 years of microbial synthetic biology to build things like biosensors for some of the carbon compounds that plants are secreting. So we could potentially have a really rapid readout of something like fluorescence based on what the microbes are perceiving in their environment if they're growing on a plant. We can also metabolically engineer these microbes to maybe use carbon that plants are giving them differentially to have the output be something like growth. And so with things like fluorescence or maybe the composition of microbes that are growing, I think this can be a really enabling platform to rapidly prototype synthetic plant constructs. And I'll go into a little detail with some of the different types of avenues of inquiry we've explored with these type of approaches. So I'm going to give an overview of the rest of my talk today. So I'm going to first talk about one main carbon source to this talk, and that's sucrose that plants are releasing. And so I'll describe the construction of microbial biosensors for plant-released sucrose and how we can use those to study the microbes' interaction on sucrose-releasing plants. I'll then go into detail about how we can use these biosensors to phenotype transgenic plants, where we can systematically dial gene expression on the plant side to dial the release of sucrose, which our biosensors can rapidly phenotype. And then I don't have time to talk about it today, but we've also done a little bit of work of engineering microbes to use sucrose if they don't normally use it. And we're beginning to understand how we could metabolically engineer the microbiome to use certain resources or not, and how that affects the fitness of these microbes with a mind towards maybe making biofertilizers or bioinoculants that can be more stable and efficacious in their deployment in a root or soil system. So the talk today will focus on the first two aspects of this research. Okay, so I talked about sucrose as the carbon source that we're zooming in on, but why sucrose? Why is that something that we may want to try to engineer and understand? Well, if you're a plant, sucrose is actually probably the most important molecule that you're making. So plants capture carbon through photosynthesis. The end product of photosynthesis is sucrose, table sugar. And so in the leaf tissues, which are the main photosynthetic centers of the plant, they're producing high quantities of sucrose. And then these source locations of the plant will send sucrose to the organs that have very high demand for sucrose, but maybe aren't as photosynthetically active. So think of this as being sink tissues like developing flowers and seeds. They have very high carbon demands, and so sucrose is sent long distance from the leaves to the floral organs to meet those biomass demands. But also sucrose is sent below ground to the root systems, which are not exposed to the sun by and large. And the root systems will receive that sucrose and use it to fuel the growth of new roots. And so over the course of a lot of plant biology investigations, people have understood pretty well the genes and physiologies that drive the movement of sucrose inside plant tissues. And so that's kind of a nice basis if we want to understand how it's being released. We have all the understanding of how sucrose is moving around from cell to cell within plant tissues. But as it turns out, sucrose is also secreted from root tissues quite prevalently. But it's less understood what are the genetic underpinnings of this process. But what we do know is that maybe somewhere as much as 10% of the root exudates, the compounds that roots secrete into the soil system, can be things like sucrose, and that this sucrose can serve as a very ready carbon source for many microbes that can break down sucrose and use it to fuel their own biomass growth demands. And so together, this kind of motivated the idea, well, maybe sucrose would be a great model metabolite to try to dissect the exchange of sucrose between plants and microbes. Could we use sucrose to understand what tools we can create to measure and control its exchange between these two kingdoms of life? So, okay, sucrose is our model metabolite, but what's our model microbe to understand these processes? So Pseudomonas petita was the one I landed on pretty early, and for a number of reasons. Folks at the Department of Energy and many other industry-facing organizations have really started to see P. petita as being E. coli 2.0. It has a lot of the attractive features of something like E. coli in that it grows pretty fast in common growth media conditions. You can do genetic manipulations in it pretty readily. And it has a lot of other, as I like to say, superpowers that maybe make it a little bit more attractive for a lot of these different technologies. So it can withstand a lot of environmental stressors. It can be used in bioremediation platforms. It accumulates a lot of different useful bioproducts like biopolymers to pretty high amounts. But most important for my questions was that it colonizes root tissues, very diverse plant tissues, very, very well. So here's a picture of Pseudomonas petita that's expressing a fluorescent protein M scarlet. And you can see that it's colonizing the roots of Arabidopsis thaliana pretty distinctly. So at the root tip, we can see the red accumulation of the protein in the bacteria very broadly. And then I'm not showing all the way up to the leaf tissues, but this red that is also from the bacteria gets even more intense as you get towards the shoot. So the bacteria colonizes very well, which makes it maybe a nice chassis to deploy genetic circuitry into, where we could maybe try to dissect what this microbe is perceiving in the environment and what it's able to consume. And so in my work, I've developed a number of broad host range plasmids to very rapidly deploy in these bacteria different sensing functions or actuation functions for things like sucrose metabolism. And so altogether, P. petita is just a really great chassis to try to do rhizosphere engineering type work. But one of the kind of also just happenstance benefits of the common strain of P. petita that a lot of folks use is that it natively does not metabolize sucrose. And so if we're trying to dissect how plants release sucrose and maybe build things like biosensors to perceive sucrose, P. petita is kind of a nice blank slate to layer on those functions and learn by design for how this microbe and plant are exchanging sucrose back and forth if we build things like sucrose sensors or new sucrose metabolisms in P. petita. So I mentioned that spatiotemporal understanding of carbon release is kind of what's lacking at the moment. But I don't want to be mistaken that people have not understood what plants are secreting at all. And through the work of folks at the Department of Energy, like the Joint Genome Institute, like where Axel's from, people have done really awesome studies of these swath of metabolites that plants are secreting under different growth conditions. And so what they can do is they can grow plants, like here they tested Avena barbata. They can basically just dunk the roots in a solution that can be collected for metabolomics. And then like they show here, they can analyze under different growth conditions or different timescales what metabolites can we detect with mass spec. And one of the things that they found for this grass, as well as in other studies for Arabidopsis and other model plants, is that sucrose seems to be one of the most prevalent metabolites at early timescales of plant development. So that's shown here in red. Meaning that sucrose is very released perhaps quite early and maybe dials back as the plants mature. But it certainly seems to be a molecule that maybe would be worth investigating. Why is sucrose released so early? How does that affect the microbiome structure? Now, the way that these experiments are done is that you have the roots dangling in some liquid soup, and you can do really awesome metabolomics measurements to understand, well, what are the possible metabolites that are secreted? But one thing you lose is the spatial resolution of where do these metabolites come from. So when these types of measurements are done, you can measure the concentration of these metabolites, but you don't know if that molecule of sucrose came from, say, the root tip, or the more mature parts of the root system, which differs from, say, plants grown in a soil system like most of our crops, where there are spatial stratification of the different parts of the plant and likely the metabolites that are coming out of these different parts of the plant. So we don't understand what are the gradients of metabolites for things even like sucrose, and we need a way to be able to measure that. And so one solution I thought was that perhaps microbial biosensors could be the answer to addressing this challenge. So the last 25 years of synthetic biology have showed us how to build things like bacterial biosensors super, super readily. So if you open any issue of ACS synthetic biology, you'll probably see a graph that looks something like this, where you have on the x-axis some metabolite of interest that folks want to try to quantify, and if you express the right combinations of biosensor genes like transcription factors, you can get very characteristic what we call response functions, where the output is something like GFP or some other reporter protein. And so people have gotten really good at optimizing these types of circuits, and we have pretty well characterized design rules for how to make a really good biosensor for pretty much any metabolite of interest. What I thought is that perhaps we could create a sucrose biosensor, which really hadn't been reported in the literature, to try to understand what are the spatial patterns of sucrose release along root systems if your microbe of interest, maybe Pipotida, is able to colonize these roots very, very broadly. So the hypothesis was that we could build a sucrose biosensor and then try to make maps where we say maybe along the distance of the root tip, what is the concentration of sucrose that our biosensor is perceiving? So to build this type of biosensor, I bioinformatically mined for a few different key parts that were needed for the microbe to uptake and perceive sucrose. And so looking in some relatives in Pipotida and other bacteria that are known to colonize plants, I was able to find sucrose transporter proteins that we could express in Pipotida. And then there had been some literature evidence of transcription factors that regulate how other bacteria are perceiving sucrose such that they can then regulate the genes that eat sucrose. And so guided by those studies, I was able to cobble these genes together and express them on broad host range plasmids to ask if the output is GFP, could these genes enable the Pipotida strain to uptake and sense GFP in the presence of, or sense sucrose with the output being GFP. And so we tried to make response functions that look like what I was mentioning in the last slide, where we can dial the concentration of sucrose in the media, and then we get a very nice fluorescence output on the y-axis. And so we can fit this data to very characteristic gene expression models. So the activating Hill function is what I'm showing on top. And we can optimize our sensors on some key parameters that are common in the biosensor literature. So we looked at mainly the dynamic range. What is the fold on versus off for our biosensors in the presence of sucrose, as well as the what's called the sensing threshold, or K1/2 parameter, which more or less characterizes what is the sensing window for, in our case, sucrose, that we can actually use our biosensor to detect sucrose. And so we would cobble these genes together on broad host range plasmids, the output for the biosensor being GFP. We also had an internal control that consistently expressed a red fluorescent protein, M scarlet. We would transform these plasmids into Pipotida with electroporation. And then with a very high throughput 96-well plate assay, we could dial in different concentrations of sucrose into the bacterial growth media, and then with a plate reader, characterize the output fluorescence of our different strains. Okay, so I'm going to walk through how we built our first generation sensor and what some of the controls were. So what we first started with was an output promoter that had what we thought was a putative binding site for one of these sucrose binding transcription factors. And so our promoter that did not have the sensor system attached to it, but was effectively a such promoter with sucrose binding TF site, was able to output GFP to a pretty high degree in our assay, especially relative to the negative control that did not have this output promoter. And so you can see just this is our negative control in gray, our positive control in purple. We then asked, well, if we add in the sensing machinery, what happens when there's no sucrose? And so these sensors are actually repressor proteins. And so when we express the sensor repressor with the transporter proteins as well, this turns off the output promoter. And we can see that with no sucrose, the signal goes down to basically the baseline. The sensor is turning off gene expression. But when we started adding in sucrose to the bacterial growth media, we saw a very characteristic sigmoidal response function pop out. And so this showed that our initial sensor design was able to activate transcription within what is the low micromolar range of sucrose, which was quite relevant to the root environment. So other studies had estimated that the root environment had a low micromolar window of environmental sucrose. We seem to have been able to build a biosensor that could detect sucrose in those ranges. So that was good first sign that, hey, maybe these could work in a plant environment. So in our first generation sensor, we tested one promoter, but we said, okay, can we make some optimizations to this sensor architecture? So in our initial optimizations, we asked, well, could we change the promoter structure to increase the dynamic range? So we went from having 2 transcription factor binding sites just down to 1, and that seemed to have a increase in the fold turn on from roughly 11-fold up to almost 40-fold with our biosensor. We found, though, that one of the most impactful optimizations was actually changing the transporters. So I'm showing you the same data here, just colored differently. Both of these these promoter constructs were using the same transporters in the backbone of the circuit. And it was bioinformatically predicted that these would have a relatively low affinity for sucrose in the millimolar range. We found that when we searched for other transporters that had a higher affinity for sucrose in the micromolar range, that led to an even higher fold turn on. And so this suggests to us that perhaps our sensor was not being fully activated initially, that maybe having a higher affinity transport could let in more sucrose to the intracellular space of the bacterium, and then that could turn on our sensors even greater. So that was pretty exciting to see that we got an almost 100-fold turn on with our optimal transporters that were being expressed. And then the last thing we did was we tested new transcription factor genes themselves. So these are what's actually binding to sucrose and then modulating their binding to the cognate promoter that's regulating GFP expression. And we found that when we swapped to a different transcription factor, that we found a somewhat higher sensing window. So with these, we're using our new optimized sucrose transporters. We're just simply swapping the repressor gene that's being expressed, as well as the promoter slightly, to be able to recognize these different transcription factors. And we found that we could now have 2 windows of sensing sucrose, what we call it our low threshold transcription factor and our high threshold transcription factor that senses about 10-fold higher concentrations of sucrose. So armed with this, we had kind of 2 optimal biosensor systems to be able to detect sucrose within the plant environment. So the last thing we wanted to test was, well, are these sensors sensing sucrose, or do they just promiscuously sense other plant-relevant metabolites? And so we tested our biosensors, both the low and high threshold ones, against a panel of other root environment-relevant carbon sources, sugar specifically. And we found that other sugars that are the derivatives of sucrose breakdown, namely fructose and glucose, as well as other disaccharide sugars like trehalose, do not activate the biosensor. So our biosensors seem to be pretty specific to sucrose and not other sugars that the plants might be secreting in certainly non-zero quantities. So that kind of gave us further validation that maybe these biosensors would be pretty specific for sucrose in the plant environment. So that's what we wanted to ask, was we had our biosensors. All I was showing you in the past was testing the biosensors in a minimal media that had these target sugars. But what about if we add them in planta, where the plants are the ones that are releasing sugar to the microbes' extracellular space? So leaf tissues, we thought, were probably going to be where the highest concentrations of sucrose are. It's actually, I think, been quantified to be in the millimolar range, so certainly above the saturating threshold for our biosensors. And then the root system is where it was still a little bit of a question mark, but we thought that perhaps we would be able to observe gradients of sucrose release. So we tested our biosensors in these 2 different contexts. So to test in-leaf tissues, we use Nicotiana benthopiana. Folks call it tobacco. It's in the tobacco family. And so tobacco is kind of a high-throughput chassis in the plant synthetic biology world. You can actually infiltrate the leaves with transgenic DNA delivering strains of agrobacterium, which I'll talk about in a little bit. And you can engineer plant cells quite rapidly. We kind of adapted that assay to inject our biosensor strains into these wild-type plants. And we asked, do our biosensors turn on relative to pre-growth conditions with no sucrose? And we could simply basically just take our little spots where we've injected our bacteria, make little hole punches, and then put these leaf tissues that had our biosensors integrated into them directly into 96-well plates and quantify the fluorescence to see, well, are our biosensors turning on relative to controls? And so here's the results of these experiments, where on the y-axis we have the fluorescent ratio of GFP to M scarlet. On the x-axis, the different treatments. And so in the open boxes, I'm showing you where we had the pre-growth of minimal media with no sucrose. And then in green is the actual leaf discs where they had the infiltration of the biosensor strains. And we actually took these measurements in the same exact plate reader with the same exact plate reader settings. And what was kind of nice was that there was no statistical difference between our minimal media controls and the strains that were in the leaf tissue for both the negative and positive controls. So the negative control has no output promoter. The concentrative promoter is the positive control. And the fact that they were the same showed us that we could make this apples-to-apples comparison. The cool thing was, though, that if we look at our biosensor strains on the far right here, the two rightmost columns, that there was a huge fold turn-on relative to the minimal media control for the tobacco leaf infiltrated samples. And so this suggested that the sucrose that was only coming from the plant was able to turn on our biosensors, and that they could work within the plant environment. Okay, so that was in leaves where sucrose is high, but what about in root systems where it was a little bit less clear what the concentrations would be? So to do these experiments, what we did was we were able to grow the model plant Arabidopsis thaliana in a petri dish scale setup where we can have a very defined media for the plants. And in this media, we're omitting any carbon sources except from what the plant is secreting. And after the plant's grown for some time, we can add our biosensor strains directly inoculated onto the root tip. As the roots grow further, the bacteria will colonize very readily. And then we can take a measurement with of bacterial fluorescence with petri dish scale fluorescence microscopy. So in this graph, what I'm showing you on the x-axis is the fluorescence intensity of the bacteria, and on the y-axis is distance from the root tip. So as you go from 0 towards the higher values, that's going from the root tip towards the leaves more or less along the root system. And so in gray, I'm showing you the negative control, and so you can see it's pretty close to 0, pretty close to baseline. The positive control is shown in purple. That's the just the constitutive output strain. So we see a shifted, but also relatively constant value of this output from this strain's plasmid. However, when we look at the biosensor, we're starting to look at the low threshold biosensor here, we do see an increase in biosensor activity as we go from the root tip towards the more mature parts of the plant. We also tested the high threshold biosensor, which, just as a reminder, is detecting higher concentrations of sucrose. It needs higher concentrations to be able to kick on. And we see that with that strain, it's only at much farther parts along the root do we start to see a separation relative to the negative control, which would make sense if the low threshold biosensor senses lower concentrations first. It's increasing as you go up the root. And eventually, the high threshold biosensor is able to perceive those turn-on concentrations of sucrose, and it also eventually goes up as well. So altogether, this suggested that the root tip has this low concentration of sucrose, and as you move towards the photosynthetic parts of the plant, the leaf tissues, the root system seems to be secreting more sucrose in that environment. So here's actually what that looks like when you look at a raw image of the biosensor overlaying the red signal and the green signal. So I'm showing you here the low threshold biosensor inoculated onto Arabidopsis. There's actually not a picture at all of Arabidopsis shown in this image. It's simply the fluorescence of the bacteria. And so hopefully you can appreciate that the bacteria colonizes super, super broadly along the root system. It basically lights up where the plant is. But we can see it, like I mentioned, at the root tip, there's not much green. You only see the magenta, which is the internal control signal from the biosensor strain. And then the green pops up more towards the mature root tissue, towards the leaves. And so, given that there was this depletion of sucrose release that our biosensor was detecting at the root tip, I thought that maybe there was a bit of a synthetic biology opportunity here to ask, well, could we tune the release of sucrose at the root tip with genetic circuits in the plant? Might this be a strategy to control the release of carbon into the environment and maybe toggle how other microbes that use sucrose are able to assemble in the vicinity of the root tip region? And so if we wanted to use plant synthetic biology approaches to boost sucrose release from the root tip, we had to ask, well, what genes could we control in that vicinity? And what tissue types exactly do we want to target to actuate the release of sucrose? So we thought in terms of genes to control that we could start with some known plant proteins that are involved in the transport of sucrose in leaf tissue specifically. So the SWEET11 transporter is a super canonical plant sucrose transporter that's very heavily expressed in plant leaf cells. And what happens is that as the leaf cells are undergoing photosynthesis, the plants will actually secrete the sucrose from inside the plant cells to the outside. It'll travel along the extracellular space of the plant leaves, and then it gets loaded into the vasculature of the plant, or then gets sent to the different organs that require it. So it's known that if you express these SWEET11 sucrose transporters, you can secrete more sucrose in leaf tissues. We thought that maybe we could express these transporters in a synthetic sense, where if we dial the expression of sucrose transporter levels quantitatively, we could thus get a dialed release of sucrose from tissues that we're targeting. And so to do that, we used a circuit known as the buffer gate circuit that was developed in my postdoc lab at Stanford by Jen Brophy, who's now also a professor at Stanford. And what we could do is we could use different architectures of the circuit where, depending on the number of binding sites in the promoter, that can affect the expression of the downstream gene, which in our case was SWEET11. So the hypothesis was, if you express more of this gene with the different buffer gates, you could potentially get more sucrose release. We wanted to ask if we could use our biosensors to phenotype these different transgenic plant constructs. So the question was, if you express more SWEET11, do you get more sucrose release? If we have our P. petita sucrose biosensors in the mix, would we see a concomitant increase in the biosensor signal? So to prototype these types of synthetic plant circuits, we used the leaf infiltration assay that we previously had tested. And so now what we could do is we could use a strain of bacteria known as Agrobacterium, which can deliver the buffer gate circuit into tobacco leaf cells. That will deliver the DNA to the plants to then express that DNA to toggle SWEET11 expression. In those same spots that we've created different transgenic plant tissues, we can then infiltrate our biosensors to ask, well, does the different transgenic tissues lead to different biosensor signals? And we could, as we did before, take hole punches of these different spots and measure the fluorescence of the plant tissue in our plate reader to quantify the different changes we might observe. So as a way to track the expression of the plant gene, we translationally fuse the SWEET11 protein with a blue fluorescent protein, M-turquoise-2. And you can see here that it lights up these pavement cells in leaf tissue. So we get a very nice quantification of SWEET11 expression just by quantifying the amount of blue fluorescence we see. And so you can see here that with each of our 7 tested or assembled synthetic plant constructs, we get this dose-dependent increase in plant gene expression depending on the promoter architecture that we're using in our buffer gate. So we can dial plant gene expression, and we again tested both the low threshold and high threshold biosensor separately into these different transgenic plant spots. So now in the bottom, I'm showing you the actual green and red signal for the biosensors that were in these different spots. So for the transgenic plant tissues that had the low threshold biosensor, again, they sensed the lower concentrations of sucrose to turn on. They were on and saturated across all our tested plant circuits. So basically, the sucrose that was being released into the environment of the P. putida strains was saturating the signal. All our biosensors on across all the tested plant constructs. However, when we compare the high threshold biosensor, which requires a higher concentration of sucrose to kick on the biosensor output, we did see a concomitant increase in biosensor output based on the plant construct we use. So together, this seemed to suggest that we could toggle plant gene expression and use our biosensors to phenotype the success of our plant circuitry, that our biosensor signal was increasing with increasing, in this case, sucrose transporter expression. So this was all in leaf tissue and at least gave us a pretty rapid output to say, hey, we could potentially use these circuits as we intended. But we really wanted to try these in a root system where, again, at the root tip was where sucrose levels are quite low for wild type plants. And so now that we had a gene target that we wanted to express, we had to be able to express that plant protein in exactly the region of the plant that we wanted. And so to get that spatial control of gene expression, we looked for plant promoters that were very active almost exclusively in the root tip vicinity. So the PIN2 promoter, or ProPIN2, is known to drive oxygen transporter expression within the epidermal and cortical cells of the root tip. And so we said, well, maybe we could reconfigure our buffer gate circuit to now drive our synthetic transcription factor that in turn activates the other part of the buffer gate, and we could drive its expression with this ProPIN2 promoter. So the PIN2 promoter controls the spatial expression of the buffer gate. We then build the 7 different variants of the different magnitude strength promoters to quantitatively dial the Sweet 11 expression at that restricted region of the root tip. So we wanted to ask, okay, we could have these 7 different buffer gate lines, the driving expression of the root tip with different gene expression magnitudes. Does this lead to a staggered release of sucrose export at the root tip as determined by our biosensors? So to build these stable transgenic lines, we again used agrobacterium to deliver DNA to plants. And in this case, we moved from tobacco leaves to Arabidopsis, which we wanted to interrogate. You can do this method known as floral dip, which allows you to basically just dunk the flowers of the plant in a soup of your DNA-carrying agrobacterium. And then you'll eventually get transgenic seeds after a few months that you can select. We grew those up on our Petri dish assays as before and added our biosensors, and then after some more time quantified the fluorescence output of our biosensor strains. Okay, so I'm going to give a recap here of what we saw with the wild type Arabidopsis, the cold 0 lines it's termed. So in the top row, I'll show you the images from the low threshold biosensor inoculated plant lines, and on the bottom is the high threshold biosensor inoculated plant lines. So again, in magenta is the constitutive signal of the bacteria. We don't see any green. The biosensor is off at the root tips of wild type plants. But what happens if we start testing our different transgenic plant lines? So now every column is a different transgenic plant line, and we're starting to increase plant gene expression. What we found was, as we started to dial up the magnitude of gene expression for our transgenic plant lines, we saw that the first strain to light up was our low threshold biosensor. So it seems to be able to perceive the levels of sucrose that our engineered plants are now putting out into the root tip environment, whereas our high threshold biosensor still stayed off. And it's only at higher plant gene expression magnitudes that the high threshold biosensor eventually kicked on. So together, this seemed to suggest that we are actually able to create a gradient of sucrose export. And this was really validated when we looked across independent transgenic lines. So a devil in the detail for plant SynBio is that when you make transgenic plants, more or less every transgenic line you're screening has that circuit DNA randomly inserted into the genome of the plant. And so depending on the context of where it was integrated, you might get just variation in gene expression overall. And so it helps you to screen across many, many different transgenic plant lines. And that's what we did here. Every dot is a different transgenic plant lines that were phenotyping in their biosensors. And excitingly, regardless of where these integrations happen in the genome, our different transgenic plant lines show the same overall phenotypes based on more the architecture of our circuit. So relative to the wild type plant, we can get this general increase in exudation depending on the expression strength of our sucrose export circuit that the buffer gate enabled. So that was pretty exciting. One thing we did notice, though, was that if we look macroscopically at the growth of the plant, we did see some things changing. And so if we analyze the length of the primary root from where we inoculated, shown here, where the marker is labeling the plants, these are the Petri dish grown plants, we can quantify the distance from where we inoculated to the root tip as some proxy for biomass formation of the plant, so the primary root length. And if we quantify that as a function of the different transgenic lines we tested, we saw this inverse relationship where the more we drive the expression of this SWEET11 transporter gene, our plants grow less. They seem to be having a shorter root system. So this was suggesting that maybe as we release more sucrose into the environment, maybe there's less sucrose that the plants themselves can use to fuel their biomass demands. There's a bit of a trade-off. We also had a control where for our strongest buffer gate line, we didn't express the plant sucrose transporter, but simply just a dummy gene, the M turquoise blue fluorescent protein. And even for that one, there was this stunting of root growth relative to the wild type control. So altogether, this seemed to suggest that just carrying this genetic circuit might be imposing a burden on the plant. So my background is more in microbial synthetic biology, and I very much saw anytime I express even something as simple as GFP in bacteria, the more you express your foreign protein, the slower the growth rate of your bacteria are. I think this might be a little bit of an underappreciated aspect that's also true in plant synthetic biology that we need to consider as we go about creating these different synthetic plant constructs. That how is the effect of our circuit outputs, maybe it's in this case the sucrose exporter, or maybe just the circuit itself that is siphoning transcription and translation resources in terms of the effect on the overall plant physiology, or in this case, the growth of the root. Okay, so just to sum up the talk and hopefully give some takeaways. So I think maybe I've shown that we can build biosensors for things like sucrose in the plant environment, and that these biosensors can be really powerful tools to phenotype transgenic plant constructs where we're, in this case, dialing the release of that same metabolite biosensors can perceive. I didn't have a chance to talk about it today, but we've also been able to successfully build strains of P. petita that can consume sucrose, and we're trying to understand how the ability to use sucrose as a growth resource can affect the fitness of these strains in a competitive microbiome environment. And so we use sucrose here as our model metabolite, but folks in my lab are now excited to try expanding this biosensor phenotyping approach to other major plant metabolites that are secreted into the root environment, but also into the above ground tissues of the plant as well. So with that, I'd like to acknowledge a lot of folks from my new lab who are taking the next steps of this project and building new biosensors and new transgenic plants. I'd also like to thank the Denny Lab at Stanford, who hosted me for my postdoc, and also give a shout out to Taylor Clark, who's research tech there, who's finishing up some of this work before we hope to submit in the coming months. I want to give a shout out to the funding sources. So the Tomcat Center at Stanford supported this work financially, and we're very excited by being able to engineer plant microbe processes for sustainability. And I also want to thank JGI that Axel is a part of, as we do have a CSP grant to quantify sucrose in the root environment with some of our different transgenic constructs. So with that, I'm happy to take any questions.
Fantastic. Thank you. This is amazing. I want to ask Axel whether he has any question or comment before I'm asking a lot of questions to you.
Okay, yeah, no, this was awesome. This is really fantastic work, quite fascinating to see the utility of these tools and just trying to imagine what you can use them for. One thing that I wanted to point out, coming back to my introduction talk, is that in fact, like the Pseudomonas putida strain that you mentioned, which is the KT2440, this is one, in fact, where we have this CRAGE technology available where you can actually engineer the genome quite easily. But that just as an aside. One question that I had, which is a more fundamental one, but might be important here. So, you clearly showed in your last few slides that if you tweak the exudation of sucrose in the plant and dial it up way high, that you see a burden on the plant phenotype. This makes me wonder. So, and I'm wondering if you have any insights here, like the basic level that we see in plants of exudation. Do you feel like this is something that the plant carefully and actively dials in by targeted release of sucrose at a certain level, which presumably is also like a smaller burden, but might be outweighed by beneficial effects from recruiting microbes? Or is the base state that the plant essentially tries to minimize what comes out, and what we see is essentially leaking out, which and the plant through, well, I would imagine through cell type specific expression of these transporters, which are of course required for, these are the same transporters that are required for normally moving the sucrose around. That is it. What essentially it's like, is it an actively managed process, or is the plant just trying to minimize it? Any thoughts?
Yeah, that's great questions. So I didn't share the data here, but we have tried other promoter constructs that do not target the root tip. And I don't recall the output of those results exactly, but I want to say that there wasn't as much of a stunting of root growth. So it might be that the reason the root tip is so depleted in exuding sucrose is because that's maybe the critical part where it needs to be used for plant growth. So maybe that would suggest that there is some bit of active regulation of not secreting it, because it's needed most critically at the meristem of the root, which is kind of where all the cell division is occurring. We're not, that's a little bit kind of hand-wavy and speculative, but we are doing some follow-up experiments to try to profile these different parts of the root where we do see kind of this staggered increase in sucrose release, doing transcriptomics to say, well, what genes are being expressed here on the plant side? Does it make sense in that maybe we're seeing more sucrose transporters actively upregulated there, or maybe repressed at the root tip? And we've also seen, I'd say, we've been testing many other plants outside of Arabidopsis, and you get a lot of different sucrose exudation patterns. And so we've seen for some plants that there's almost like no sucrose release at all. So my feeling is that it probably is actively regulated. I think I've heard people say, well, carbon's cheap. But if you're a plant, and that's like one of the most important molecules that you make, I would imagine you probably want to control when you're putting it out into the environment. But yeah, we're still very actively probing that.
Yeah, I mean, well, it's cheap, but it's still a cost. Also, I would imagine that you have to think about like beneficial versus non-beneficial microbes that you might attract if you just put too much carbon out there as a food source, right? In a like outside the well-controlled system that you use.
Mm.
Yeah. Another comment that or question that I had was, so I was quite excited to see how you're engineering both the microbial side of things and then the plant side of things. And look, like so far you're engineering these in isolation, but of course, in principle, there is the potentially very exciting possibility to like do targeted engineering of reciprocal communication loops, right? I mean, so right now you are controlling the expression in the root, but in principle, you could envision a mechanism where, depending on the sucrose concentration, the microbe produces some kind of communication molecule that get sensed by the plants and then in turn can modulate the strength of your expression. Have you thought about that at all?
Oh yeah, definitely. Because I think we just kind of initially want to ask for primary metabolites that are secreted. They are probably by and large the majority of foodstuffs if you're a microbe below ground. They likely, and has been shown in other studies, have a huge effect on the microbiome composition. You have more sugar consumers if there's more sugar in the root exudates versus, I say, organic acids. But to your point, I guess, if we want to engineer like a plant growth promoting bacterium to maybe use sucrose on a plant that we've transgenically created to release more sucrose, you're going to get a lot of cheaters that maybe aren't the target bacteria, but can use that resource. So we've been thinking of ways to, as we say, like insulate that carbon source to something that's maybe more specific to your target microbe. And I think, as you kind of alluded to, maybe make it more of a signal to friend, hey, I'm here. Now it's the right time to release this carbon. So there's a few cool studies I've seen where people do kind of this trans-kingdom signaling, where it's not really a molecule for consumption, but more just perception of a signal. And I think we have, in some of our later experiments, we've been interested in, well, can we trigger the release of sucrose with maybe one of these types of signaling modalities?
Yeah, that sounds great.
So do you have any other question or comment?
No, that's it from my end.
Sure. So let me have two comment or question. So it's kind of related to what just Axel just mentioned. Actually, the first question is kind of one thing I also want to ask, but the second part of his question is about bidirectional communication. One thing potentially we could discuss later on. We recently developed all kind of auxin sensor, including auxin derivative. And then we, able to, engineer bacteria to produce auxin and auxin derivative in the future. And then that potentially, allow, people to make that bidirectional communication in the future. And then we have the specific sensor for auxin derivative, and then they can also use for your kind of experiment in the future. So that's an exciting area, and I'm happy to chat later on. Now, the question I have was, to me, your choice of P. putida is very smart, because my naive knowledge of bacteria basically also have chemotaxis, but I believe given P. putida doesn't have sucrose sensor originally, that means probably this putida probably not gonna attract it toward the sucrose gradient or whatever high concentration sucrose. That means the distribution is without any those kind of chemotaxis, probably distribution along the line of the root basically the probably similar or the identical across that one. But if you choose some other bacteria, for example, I guess if that attracted toward the sucrose, you have the uneven distribution in terms of the population. In that case, I mean your analysis might be a little bit challenging because you need to kind of compare two color in terms of the distribution. So I don't know. I mean, is it true? I mean, in terms of population distribution for other bacteria that has the chemotaxis, or this is not known to the community yet?
Yeah, I mean, it's a great question. And that's very much why I chose P. putida was because it had, like you say, kind of that it's kind of blind to sucrose.
Mm-hmm.
And it just is super broadly colonizing. Definitely, it's known that other root colonizing bacteria do have very preferred niches along the root. So,
Yeah.
another bacteria that I didn't work with too much, but others in my postdoc lab used was a bacillus strain, and it very preferentially colonizes the root tip. So there, maybe you could sense root sucrose at the root tip, but maybe not elsewhere. The way we built these plasmids that I think I mentioned during the talk was that they're all on broad host range backbones. So we have a huge collection of rhizosphere isolates, genome sequence and not that we're shooting these biosensor plasmids into, and we're trying to see if the patterns that we're observing are broadly true depending on what bacteria that's colonizing. So maybe they colonize in different spots and you kind of get this mosaic of colonization, but does the biosensor signal all show the same thing? It's just this general increase. We don't exactly have the answer to that just yet, but we are very interested in just, and these are, I should also say, these are all single strain studies. In a whole microbiome context, we have tons of different bacteria. What are the patterns? Are they what we're observing, or is it something different? So it's a good question, and how would we look at other metabolites that are being consumed? I think maybe one answer could be using different microbes that could be best suited to quantify these metabolites, but also just understand how they're changing in a more realistic mixed community setting.
Well, that makes sense. Thank you. I now see, we already passed, 11 Central Time, almost 20 right now. So probably we close and then we could chat more after we close. Okay, so thank you all for joining and staying today. And we will meet again on March 20, Thursday, the same time, the same Zoom link. We'll have Professor Simpson Belkin at Hebrew University of Jerusalem, and also Dr. Myung-hyun No at Korea Research Institute of Chemical Technology. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I stop recording. Just give me 1 second. And thank you all again. Just give me 1 second.